Good morning, good afternoon, or good evening, depending on where you are in the world. And welcome to today's QMed Focus on Fundamentals webinar concerning guidance for changes in a medical device, sponsored by Nelson Labs and broadcast by Informa. It's day one, and we'll be discussing ethylene oxide and radiation sterilization. I'm Bob Sperber, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, share this webinar, and participate in the Q&A session that takes place at the end of our presentation. The slides will advance automatically throughout the event. Also, if you haven't already, be sure to click the Register Now button in your viewing window and join us for the remaining 2 days of this Focus on Fundamentals series, where we'll be discussing packaging, biocompatibility, and biosafety. Lastly, if you're experiencing any technical difficulties, please click the Help widget found at the bottom of your screen, or type your issue into the Q&A area, as well as questions, if you'd like, and we'll be glad to offer one-on-one assistance. Now, on to the day one presentation, ethylene oxide and radiation sterilization, evaluating the impact of a change. Discussing today's topic is Gregory Grams, Sterilization Technical Director at Sotera Health Expert Advisory Services, and Bryce Telford, Expert Technical Consultant at Nelson Labs. To learn about our speakers, visit the BIOS widget on your screen. First off, Bryce, over to you. Are you there, Bryce? Oh, I bet you're on mute. This just shows how we're live. We'll get with you in any case. This is one of those things that happens very rarely, but it's nice to see. So I can ad-lib, but you already know why you're here.
Okay, can you hear me?
There you go. I was running out of thanks, Bryce.
I don't know why it did that. Oh well. Sorry about that, everyone. So my name is Bryce Telford. As mentioned, I'm with Nelson Laboratories. So today we will be discussing the effects of change. So for my presentation, I'm going to discuss some of the biggest changes we've been seeing in the last 2 years, and that really has to do with environmental or safety changes when it comes to radiation sterilization. So when we talk about environmental changes and safety changes, we all had and were impacted by the experience of COVID. So that led to many changes, not just in our daily lives, but also in the manufacturing of our products. In some cases, this meant an implementation of masks or face shields, gloves, gowns, but also additional environmental factors. In some cases, that meant increased or improved filtration, UV disinfection, increased surface sanitization, and even separation of shifts. And all of those changes have the potential to affect and impact the production of the product and therefore its sterilization. Anytime there is a change to production, there's a potential change to bioburden. And that potential change to bioburden, that means effects to your sterilization. So my focus in particular is radiation sterilization, which is a bioburden-based sterilization method. And anytime we have a change in bioburden, since it is a bioburden-based sterilization, that can mean a fairly large effect. But it's also going to cascade to other sterilization modalities as well, and other aspects of product safety, such as endotoxins or particulates, as an example. So I like to break it down as the good, the bad, and the ugly. So the good is when we implement PPE changes, when a lot of people added in basically items in order to protect their personnel from COVID, in a lot of cases, that also made some positive changes for the product. So let's say we have an implementation of mask wearing. Well, that has the potential to decrease particulate and bioburden contamination from the face and mouth area. Similar story with the face shield, as well as gloves and gowns. And from the environment, then maybe we're improving the building's filtration to try and basically get contaminants out of the air. COVID in a lot of cases, but it also has side effects of being able to get rid of other contaminants as well. Maybe we're implementing UV disinfection, so we're reducing environmental bioburden that's present in the first place. Also an increase in surface disinfectants, a lot more cleaning, more routinely wiping surfaces with alcohol or with Clorox, with hydrogen peroxide, in order to have a sanitary environment. And also, in order to prevent spread of COVID, there was also usually a physical separation. Sometimes that meant a separation of shifts. And there were situations that we observed where that actually led to some improvement as well. So, for example, you had clean rooms that were able to recover and have a state of rest to basically cycle the air and lead to a bit of a reduction in bioburden there. However, there was bad that went with this. So with the implementation of masks, there, for example, was a lack of mask use protocol. So that, in a lot of situations, led to relaxed aseptic and hygiene processes. So rather than sneezing out of the hood, for example, if you're working in a hood environment, people figured, well, I have a mask on, so that's going to stop the particulates, and they would just sneeze. But that had the effect of basically blowing the particulates off the outside of the mask and into the hood environment. So PPE is not a replacement for good aseptic practices. We also saw with the face shields, you didn't have direct breath on the product, but in a lot of situations, the air basically still had to flow somewhere. It had to go and do something. And in a lot of situations, that led to unexpected forms of contamination. Things weren't basically flowing where you wanted them to. And with gloves, with the implementation of that, we have what I refer to as the magic force field effect. I'm wearing gloves, therefore I can touch this and I can touch that. An example of this is if you've ever gone to a restaurant where they make the sandwich in front of you, you might have the person that they don the gloves, and then they'll scratch their head, and then they'll go right back to prepping your food because they're wearing gloves. It's okay. I have this magic force field that lets me touch anything. When in reality, the gloves are picking up all the contaminants that you're touching, and that loss of hygiene is really going to affect production. With gowns, again, we ran into issues where there was a lack of use protocol. We had issues where they weren't being cleaned or laundered on a regular basis. We introduced gowns, so we also introduced particulates that were going with those gowns. And then there was also the cost associated with any laundering or upkeep with the gowns. On the environmental, it would seem at first glance that if you're introducing filtration, the expectation is that you're going to have improved air quality. But without proper planning, without proper engineering, and without proper change control to go with it, then that can actually lead to some issues such as a reduction in the necessary airflow. So areas that should have been at positive pressure now have reduced airflow, and they're no longer at that positive pressure environment. UV disinfection, the lamps are quite costly, and they are also have a very set life to how effective they are, depending on the manufacturer. The other thing that we found with UV disinfection is it's an example of directional genetic selection in process. So things that are UV radiation resistant have a tendency to also be a bit more radiation resistant in general. So you're creating an environment that's actually selecting for radiation resistant organisms. So with that, there's also some potential negatives. So increased surface disinfection, we also saw with that increases in particulates. you're basically spraying aerosols, more cleaning materials. You're also going to see more residuals, and those residuals can then begin to build up and become an issue for the product residuals or product particulates. And then with separation of shifts, we saw disruption of basically the shifts. You didn't have a smooth transition from one shift to another. So you had all the shutdown process and all the basically startup process that was leading to additional contamination events. Now, the downright ugly was really kind of after the fact, that was a lot due to global supply chain issues. So with masks, there was implementation of the masks, and then everybody was using masks. But suddenly there was then mask shortages, or getting the proper masks became difficult. Facilities that previously were using masks, such as KN95s, suddenly weren't able to get a hold of them. So changes had to be made, implemented. So instead of a KN95 mask, they're just using regular masks. Well, that can lead to increased particulates and increased bioburden than compared to their previously controlled setting. With gloves, we saw a shortage of sterile gloves. So in some situations, to make do, you're using non-sterile gloves or gloves that you hadn't previously qualified. Those gloves may not have been vetted properly, could increase particulates. They're not sterile anymore, so now you're increasing bioburden. And then with gowns, we saw a change also with going from disposable gowns to reusable gowns. And in some situations, that was perfectly fine and acceptable. In others, it wasn't a well-vetted process, and there wasn't sufficient controls in place. So you were seeing increases in bioburden because instead of wearing a gown for one session, it's now being worn for an entire week. In other situations, that wasn't actually a bad thing because the gowns were so well controlled that they're being only handled in that controlled environment. They're not really having an opportunity in that controlled environment to pick up additional contaminants. So because of the controls that were in place, it was actually acceptable. And then when it comes to filtration, again, global supply chain shortages, unable to replace filters, unable to replace them in a timely manner, employee turnovers and difficulty hiring new employees. So there was a reduction in maintenance staff. So routine maintenance wasn't being performed in a proper, orderly manner during proper intervals. So what you were seeing is in a large variety of environments, you're seeing these changes. And the more controlled the environment it was, then in some aspects, that control basically was able to cope with those other changes. But in others, where you had a lot of change due to supply chain issues, relying on having that absolutely pristine condition, it led to a lot of issues. And on lower level clean rooms, basically in some, you actually saw improvements because you're implementing masks where you didn't have them before. And in other environments, you were seeing improvements because you had higher levels of filtration and you were doing a better job of cleaning up the environment. So it also depends and really mattered on how robust the individual change controls were and how well vetted out the process was. Unfortunately, in a lot of situations, time really wasn't on anyone's side. So a case study is one where we had a dose audit failure. And normally when we have a dose audit failure for radiation sterilization, we start looking for what changed, what's different from last time. And a knee-jerk reaction tends to be, well, nothing changed, nothing's different. However, usually as we start looking through, we start finding various changes and we can look for root causes. However, in this particular example, rather than finding the needle in the haystack, what we instead found was basically a haystack that was full of needles. So when we did the dose audit, as an example, the bioburden was within normal variation. So it was bioburden that they were used to seeing, and it was actually even below the average that was used to set the dose. The product's E-beam sterilized, so it is exposed to E-beam dosing. In order to aid with that, it was pre-manipulated product, so the product was sent to the laboratory, cut up, and then repackaged prior to dosing to facilitate proper dosing. So the certificate of processing for that dosing didn't show any issues. It showed that the dosing was within specification. So then when we did the test of sterility, we had 2 out of 10 positives. That triggers confirmation testing, which had an additional 2 out of 10 positives, indicating we had a failure of the dose audit. Now, 2 out of 10 both times shows that we're actually really close. We've just moved out of basically our sterility assurance level. We've just had that bit of an increase that's pushed us out of being able to have our sterility assurance level. So what we did, of course, if we have any failure, is we start looking at things. So we look at what changed, but we also make sure we gather the data we can. So we had the positives identified. We found out that they were spore formers, but neither organism was really known to be radiation resistant. It's not like a Deinococcus radiodurans where we can basically just point and go, aha, we have this problem. So when we ask what changed since last time, we found that they had implemented reusable masks due to COVID. However, with that, they really didn't have any procedure for those masks. You had employees wearing those masks at home, into work, on break. Those reusable masks were going into and out of the controlled environment. So the next is they had also implemented an IPA cleaning and disinfection step for their services. So they were using now isopropyl alcohol in order to sanitize. So with that, because the product is E-beam sterilized, there was also some changes in the background that we weren't entirely aware of, but the manufacturer suspected that there was an issue with either the sample preparation or the verification dosing itself. So as part of this, we actually went through and we came up with a new configuration for the product to establish a better verification dose so that it could receive a tighter dose range when verification dosing. We also improved our practices when it came to manipulating and packaging the product, adding additional layers to the product because since it was manipulated, we had a lot of sharp edges. So there was a feeling that really that was the true root cause. And we went ahead, we did a new dose after we established a new verification dose, a new dose map using a better aseptic technique and better packaging, and it passed. We had an acceptable dose audit. They also, on their end, just as a matter of process improvement, implemented protocols in order to improve their mask procedures so that they had more consistency and less basically uncontrolled bioburden entering their clean room. They made the masks a controlled process to control what was going in and going out of the clean room. However, the next dose audit came along and it was, again, below the previous bioburden. It was actually even below the previous failure. We saw a reduction in bioburden compared to what we had even seen. However, the product was considered underdosed. So with underdosed product, there is the option to proceed with your dose audit at risk. We did so at their direction, and there were 3 out of 10 positives for the test of sterility, which is an automatic failure for the dose audit. We repeated the verification dose experiment because it was underdosed. That is allowable. And that one was dosed properly and within what is considered the acceptable dose range. However, we once again had 3 out of 10 positives for the test of sterility. When we had the positives identified, this is where things got a little bit interesting, is we recovered pretty much the exact same organisms we had seen during the previous failure. So that told us, well, it wasn't really an issue with dosing or sample prep. It's probably more so we have an ongoing issue that we haven't truly addressed. So we need to look deeper. We need to look into this. And as mentioned in the previous slide, we knew that the organisms were spore formers. So when we have spore formers, we basically look at, well, why do we have spores in the first place? If it's a gram-negative, maybe we're looking at water. If it's a positive cocci, that usually tells us it's environmental and personnel. So for example, Staphylococcus epidermis, it's pretty much on every human being. But when we see positive rods, we started looking at the IPA. We started looking at the alcohol and the sanitization process. And what we found is that due to the various changes taking place and various supply chain shortages, they were using IPA, but they had to switch suppliers. They didn't really understand that that change in supplier meant that they went from sterile IPA to non-sterile IPA. However, we did do a direct test on the IPA, and no organisms were recovered. We also found that throughout the process that a single IPA container was being used repeatedly over the course of days and even weeks. So they were basically buying in bulk, and rather than using the IPA they needed and buying in small quantities, they were buying in bulk and basically going into and out of the container multiple times over the course of days and weeks, and they weren't changing out the IPA, and there really wasn't a lot of control either about getting in the IPA and handling things aseptically. Also, water baths were being cleaned weekly, and they were also using the IPA for the cleaning. They weren't using a sterilant or a sporicide. For the water baths, they were only changing them weekly, not as routinely as they would have liked, and they were using IPA for that cleaning process. So they had really started using a lot more IPA, which really was kind of doing a lot more selection to basically select for spores that could survive and even thrive following the IPA. So when we also reviewed the failures, we looked at, well, what was common between the two, but different during the rest of production. So again, we usually get the answer of, well, nothing's really changed. But as I mentioned, as we dug a little deeper, we found non-sterile IPA was really just kind of the tip of the iceberg. So what we found was in order to find out what was going on, we decided to do some in what we refer to as investigational testing. So the product was fairly complex, so we separated out the product into subcomponents, and we're basically looking at the various subcomponents to see where is the bioburden coming from, and can we find these organisms again in order to try and find what component, where is this coming in on. So within the subcomponents, we had also found that there had been some supplier changes. So one of the big changes that we noticed is that they had some natural materials in there, such as paper. And with that, they had a change of supplier. They switched from basically one supplier source for their instructions to another supplier source. And whenever you have natural materials, this is something to be very mindful of, because the bioburden on a natural material can really vary, not just from supplier to supplier, but even batch to batch and crop to crop. So that's something to be very mindful of and to certainly monitor and keep an eye on. And if possible, to eliminate. So with that, that was actually one of the things that we looked at as a corrective action, was eliminating that paper from the sterile barrier. But other items that we found were that plastic components that the supplier could no longer get a hold of. So they changed out that component. It's the same item, but from another supplier. And when we were told the same item, for example, they said, well, it's the same geometry made out of the same material, but it's from a different supplier, so we switched that. So that was also another change that we found. So really, in this case, there were a lot of changes, and narrowing down which specific change really was going to make the biggest impact was kind of an issue. So it's unfortunately a case where we really have to take a shotgun approach. So we never really did find the second organism. However, we did find the first. We found what subcomponents it was on. We found the change in paper supplier. We found the change in component supplier. So we were able to take some corrective action there. But also, there was a change from isopropyl alcohol to using a sporicidal agent when possible. That also did have to lead to some additional, basically, assessment of the product to make sure we don't have any residual issues. They also updated their water bath procedures to be cleaned on a more regular basis and to use a sporicidal agent in order to do that cleaning instead of IPA. And IPA was to have a suitable period of acceptable use. So again, I mentioned that they were going to the same container over and over again for days, weeks, and what we found out later was even months. So they made sure that they had expirations for their IPA where they figured a suitable, reasonable amount of time where they could use the same IPA bottle and it still be acceptable. So although the bioburden really remained relatively stable, we even saw reductions in bioburden. What we were really seeing was a shift in the nature of the bioburden. We were seeing increased resistance of the organisms that were present that really weren't being accounted for. So we also had additional effects, basically, that we then had to account for because of these changes. So as I mentioned, we had implementation of sporicidal agents, things like that. So that led to also having to reevaluate, well, is my biocompatibility still acceptable? Are my particulates acceptable? And since they had a dose audit failure, that meant having to use a higher sterilization dose. That led to having to assess, well, is the max dose for my product acceptable? Is it able to withstand that max dose? So really, this is an example where the focus on fundamentals, making sure that you're considering your changes, really is going to lead to a more controlled product so that you don't have additional issues. You don't have to reassess your biocompatibility. So a few items to really consider with this is the changes that can affect your product bioburden. So a product that's the same shape, made with the same material, doesn't mean that it's going to have the same bioburden. You're going to have different suppliers with different handling processes, different packaging processes, and that can all lead to basically changes in your bioburden. That also can mean changes to your biocompatibility. For example, just because one supplier says it's the same material as another doesn't mean it necessarily is, and it doesn't mean there isn't differences in the process, such as release agents or coatings that have potential impact to the product. So another item to consider is anytime you have natural materials, especially papers, cottons, things like that. Natural materials are going to vary and they should be accounted for on a regular basis. Incoming testing really is going to help ensure that you have proper control, or if you don't have incoming testing or you have the option to do so, a cleaning step, a disinfection step may be able to help. Washing the cotton, making sure that it's sanitized so that you have consistent bioburden, something in order to make sure that that natural product coming in is going to have consistent bioburden going out. So changes in suppliers, those are going to affect your bioburden, but they're also going to affect potentially your resistance. You might end up doing a bioburden test, finding you have the same amount of bioburden, but manufacturer A basically has a lot of Staph epi, manufacturer B has a lot of Deinococcus. That's going to lead to a very big effect when it comes time to do your dose audit and when you're doing your sterilization. Another thing to consider that we've been seeing a lot of is that not all water is created equal. So there's a big difference between tap water and distilled water and sterile water. And even within sterile water, there's also sterile water for injection, which basically has particulates testing with it as well. So something to consider is distilled water doesn't necessarily mean sterile water. And kind of a last note is that changes that are intended to be good, are intended to be an improvement, aren't necessarily always good if you don't have the proper training and background behind it. The example that I routinely give is the gloves and what I refer to as that magic force field effect, where I have the gloves on, so now I can touch things, I can handle things, it's okay because I have gloves. There still needs to be that understanding, there still needs to be that focus on aseptic handling, aseptic processes. So making sure that that change is understood and making sure that it is accounted for. So that's kind of the focus from a bioburden and change perspective in order to keep an eye on those items. Make sure that you're keeping things under control and some items to consider with that. So with that, I turn it over to my associate Gregory for the EO side of things.
Thank you so much, Bryce. It was really a comprehensive review of the radiation changes that may occur with the bioburden. I will now cover the other major sterilization modality, which is ethylene oxide sterilization. So unlike radiation, it is not most of the time a bioburden-based method. It is based on biological indicator resistance and deactivation. And first of all, the first question that I would have is, why would I validate a new cycle? Because if I have a legacy cycle that is working fine for a long time, I don't see a reason why I would change. In 2019, the FDA has opened the EO Challenge 1 and 2, and EO Challenge number 2 was really asking the sterilization contractors as well as manufacturers to see how we could have a sustainable ethylene oxide sterilization over time, because it is a harmful gas. It is well efficient and effective for sterilization, but control for environmental consideration is a bit more complex. And this is why Nelson Labs and Sterigenics, which are sister companies, have embraced this challenge number 2 and have started a global program a couple of years ago called the 3R of sterilization that has a main goal is to reduce the footprint of ethylene oxide, not just in our cycles, but also in the premises, in the distribution center, and in the communities. So definitely the goal is to capture all EO we have in our sterilization facilities, but also reduce the potential EO that can remain in the packaging and in the products. So how can we develop new cycles? What are the pathways that we can follow? Because there is no magic behind reduction of ethylene oxide. We cannot just say, well, I'm going to use a bit less gas and it's going to be fine. It's not going to be as simple as it is looking here. So first of all, we have to think about the cycle design. What can I change in my cycle to make it more effective? And what can I change compared to what I've been doing over the last years? A second aspect to evaluate is the PCD recalibration. PCD standing for Process Challenge Device, and I will explain that in a minute. The third aspect to consider is the validation methodology, because for the one that are familiar with ethylene oxide sterilization, half-cycle overkill approach has been broadly used. Probably 97% of the validation were following the validation methodology of the half cycle. And if I'm doing the comparison with radiation, it would have been the VDMAX 25 method that would be applied by default. But there are alternative approaches that can be used that can have better suited cycle that can be developed. Of course, if we are changing any of the parameters here, we need to assess the changes and to see what impact it might have on different aspects. So what can we modify or improve in a cycle? The vacuum is one of the critical elements, because as deep as we can go, the better penetration of gas will be. but it may have an impact on the packaging and also on the product resistance. Just giving you an example, pre-filled syringes cannot withstand deep vacuum because the plunger might be moving, and then you can have leaks in your pre-filled syringe. So it has, of course, some limitations, but we need to review these limitations to see what can be improved. Second parameter that is quite critical is the temperature. So if we can increase the temperature, because ethylene oxide sterilization is an alkylation process, so a chemical reaction, and the higher we can go in temperature, the faster the chemical reaction will take place and occur. So it means that temperature is a really key driver, and it is probably more important when we decrease ethylene oxide concentration, because we have less EO that is available to sterilize, so we need to increase the pace of the chemical reaction. Relative humidity is also a good driver. It's improving the gas diffusion process, working as a catalyst, but is also adding less resistant to the spores. It can help the spores to become vegetative and be more easy to be sterilized. And of course, we can decrease ethylene oxide concentration, but to guess or to evaluate what would be the concentration at which it will work, the advice is to do some developmental work and process definition activities, because despite the fact that we are targeting 50% reduction, sometimes it's possible to go beyond this theoretical concentration, and some successes have been recorded with decrease of 80% of ethylene oxide concentration. So it means that if we are assessing and developing the cycle with a few trials in a smaller chamber, we can know much better what are the conditions at which we can work later on. Ethylene oxide concentration is basically 4 major critical parameters. We have, of course, obviously the EO concentration that is playing a key role. The chemical reaction is taking time, so the exposure time is also one of the critical parameters. As I said previously, temperature is probably one of the most important elements to consider. And relative humidity is also one of the 4 pillars that is dictating the EO sterilization efficacy. So it means that instead of having only the dose for radiation, we have a variety of different aspects that we need to consider. It's a bit more complex to fine-tune and find the right parameters to design the best cycle, but we can see with this chart that we can play with the parameters. So if we are reducing the concentration, we can either play with a temperature, increase it, or with the time, and we have to find the best compromise. So willing to reduce EO too much can then extend the duration of the cycle, which is not the initial intention. I will now cover one important element. So we are using biological indicators to do the validation, and typically we are placing a 10 to the 6 BI in the worst case location in a product. But there are alternative methods that can be acceptable. So we can still continue to spore the device, but instead of using a 10 to the 6 BI, we could use a reduced population BI that would be more in line with the true bioburden of the device. We could do also a test of sterility and do a really graded exposure cycle without sporing the device and using PCDs. But we can also try to develop surrogate PCD that has a resistance that is more in correlation with the true bioburden of the product. In order to make sure that we can use alternative approaches for sporing up the products, the bioburden knowledge is essential. Despite the fact that we have a method that is BI-based and not bioburden-based, we can also validate an EO cycle with a bioburden approach, which is a bit different compared to what has been done before, but it is still something that is possible. But in order to be able to do that, we need to make sure that the BI that is being used is really proving that it's more resistant than the natural bioburden. And control on the bioburden is really key and essential, because I had heard several times that because we are using 10 to the 6 BIs, we can probably go away from bioburden testing, but it is not true at all, because even if we have a lower count of the bioburden, we could have still resistant organism in the bioburden. So it is still mandatory and a regulatory requirement to test the bioburden on regular basis. Now I'm giving you an example on how we can get away from what has been done before. So in my example here, we have a 6-meter PVC tubing that has been used forever as a PCD for a customer. And we are placing a 10 to the 6 BI in the center. So we cut the tubing in half. We have a connector, we place the BIs, and we have the worst case item that is covering the most difficult to sterilize location. Fine. But what would be the true bioburden in an extruded tube at that location? I guess probably close to zero. We probably have no bioburden because we have extrusion taking place at elevated temperature, and probably nothing will survive this temperature. So is it really a good idea to place 1 million spores, resistant spores, in a location where potentially there is no bioburden? Maybe this is not the best that we can do to optimize the PCD selection. So in order to still be compliant with the 11135 standards, we can still use a BI in that location, but maybe place a 10 to the 3 BI that will cover a potential bioburden, and place a 10 to the 6 BI in the primary packaging where the tubing bioburden would reside. So it means that the bioburden is likely to be more present on the outer surface of that tubing when it's rolled and placed in the primary packaging rather than inside this tubing. By doing that, then we could just confirm that the 10 to the 3 BI would be still more resistant than the natural product without artificial contamination, but that the 10 to the 6 place in the primary packaging would become the most difficult to sterilize location in the packaging. It will still be compliant with the 11135, but will of course lead to a much easier process challenge device. And with this example, we have been able to reduce the duration of the cycle without changing the concentration by 75%. So it means that we could have a quarter of a cycle being able to reach the 10-6 SAL that we were targeting for. So it means that we still have margin compared to natural bioburden, but we could reduce significantly the amount of exposure time that was required. Of course, the focus is not just on time, is also on EO, so I'll come to that point a bit later. But the advantages of an appropriate PCD selection may help to reduce cycle duration. And if we reduce the cycle duration, we can increase the capacity of the sterilization modality. So it means that the shorter the cycle can be, the more cycle we can have as a throughput. So we can have, of course, less EO being used, and if we are reducing the amount of ethylene oxide in our cycle, naturally the residual level will be also lower. So it means that we could have a faster release time to market because the heat iteration time can be shortened as well. And definitely by reducing EO, we will reduce the fugitive emission coming out of the packaging and the products and the corrugated boxes. But as I said, we are not only targeting to reduce the time. We are focusing on reduction of ethylene oxide concentration. And in the example that I have given here, if we are using the BI, the 10 to the 6 BI in the worst case location, now with a concentration at 250 milligram per liter instead of 700, we are not obtaining the kill that we had before. Of course, obviously, with much lower EO concentration, we are not getting the same lethality rate. But we are, with the same exposure time, killing the BI in the new location that is still worst case compared to the bioburden. So it means that there again, we can save time and definitely decrease the EO concentration. So finding the right balance between time, temperature, and concentration is key and essential. So To summarize the recalibration of the BI resistance and the PCD selection, we cannot get away to comparing the PCD resistance to the natural bioburden. It is still a requirement, and I encourage all of you to still do that. If we are having alternative method to store up the devices, we can still meet the 10 to the 6 BI in a, I would say, easier to sterilize location, but that would be more resistant than the worst case location natural bioburden. Bioburden characterization over time remains critical. So it means that if we are basing more our validation for ethylene oxide on the bioburden, we of course have to have control of that bioburden species and levels. But we can be innovative and create more sporing location in the product, or use a surrogate PCD that would match and exceed the resistance of the bioburden of the devices. So I have covered the PCD calibration. Now I mentioned that all of you that are familiar with ethylene oxide sterilization have been probably using the half cycle approach most of the time. But what would be the alternative that we can use instead of this half cycle approach? So the ISO 11135 Annex A is allowing to use alternative approaches such as direct enumeration or fraction negative. But still in overkill, we have the Annex B of the standards, the B12B section that is using the cycle calculation overkill approach. So it is still an overkill based on biological indicators, but that it can allow a few positive BIs, and we can not just double the time, but do some mathematical calculation to have the D value assessed, and then we can have the 10⁻⁶ by calculation. And lastly, we can use still the BI bioburden approach, in which we can define a maximum bioburden, and instead of having 12 spore lock reduction, we could live with 9 if the bioburden is limited, as an example, at 1,000 CFUs. So, of course, you may not all be familiar, and you may think that it will require much more work than the half cycle. So the half cycle is typically requiring a few fractional cycles in which you will compare the resistance of the bioburden and the BIs and the PCD. Then you need 3 half cycle, and depending if you are validating the heat iteration time, 1 to 3 full cycles. So the bare minimum quantity is 5 cycle. For the alternative approaches, so fraction negative or cycle calculation, we are not requiring much more work. So the minimum quantities is either 5 or 6. So it means in terms of execution, it will not require much more effort and cycles. Of course, the math is a bit more complex, but this is why we are helping you with our experts to do this calculation for you. So by switching from one method to the other, we can also reduce the duration of a cycle. So it means that combination of a PCD selection that is more appropriate, as well as alternative technology or alternative methodology for validation, can save a lot of time and concentration. So without changing, I would say, too much the cycle parameters. If I am changing my cycle, of course, it will bring a lot of questions. We need to evaluate whether it has an impact on the functionality of the device, because it has to be sterile, but it has still to work. So functionality is a key element to review. In terms of biosafety, if we are changing parameters, we need to make sure that we are reviewing residuals and the potential impact it can have on cytotoxicity, hemocompatibility, or genotoxicity. And for the packaging, we need to check whether the primary packaging, the sterile barrier, or having shipping and distribution validation, or what would be the shelf life. That's the new modality of sterilization will lead to. So we need to ask ourselves these questions. Of course, we need to do some homework to see what is already known with the legacy cycle and the history of the devices. So we need to review the history for product functionality, and based on the original parameters, you can define the threshold values and the maximum values for temperature, pressure, humidity, and so on, so that we know if we have margin or a bit buffer to play with. So it's important to know all the activities that were done before are still relevant, and we can review them. But if we are changing materials, or if we have changed other critical parameters that can have an impact on the bioburden or on the absorption properties, it has to be revised as well. So basically, you can use the AMI TR28 for change assessment, but basic questions can be already asked regarding the temperature. Did I decrease or increase the temperature? Because it can have an impact on the product resistance and functionality. Same for the vacuum, or if we are adding more pulses or injection in the cycle, it can have an impact on the packaging resistance as well. If we are adding more humidity, we need to make sure that we are not having liquid water during the sterilization. If we are decreasing the gas concentration and keep all the other parameters, normally we are on the safe side, but we need to evaluate if we can reduce the heat iteration. So it's still requiring to do some homework, and the duration of the cycle may be outside the originally validated window. So it is important to evaluate this as well. So the temperature changes is, of course, one of the key drivers. So the higher we can go, the better it's going to be for the cycle. But in terms of material characteristics and any adhesive that are being used in the device, we need to be cautious about elevation of temperature. So we need to find the best compromise that will leave the product functional without really impacting all biosafety and without changing the integrity of the packaging, but still be efficient for the sterilization. If we are talking about the pressure and pressure changes, I've been only focusing on the depth of the vacuum, but the rate is also important. So if we are pulling a vacuum faster, it can have also adding more stress on the packaging. So it's important to consider that as well. So I would say the closer we can stay compared to the original cycle and improve it wherever it's possible, this is where we are going to minimize the testing burden. That's definitely important to consider when a new cycle is being designed. So relative humidity, I say it's maybe some portion of the device are humidity sensitive. We could have hygroscopic powders or bioresorbable plastics that could be affected. So it has to be revised as well. And if we are increasing humidity and getting closer to the dew point, maybe we could have issues with the sterile barrier system with a paper-based, cellulose-based packaging. And finally, if we are only changing the EO concentration, as I said, there is no magic behind. It is still a pure science. So if we are reducing the EO concentration, it will require more time. So that's why it's a combination of different parameters that we need to apply together to have a successful outcome for the validation. But these changes have to be verified, and potential impact it might have on the duration of the cycle is key as well. So just to summarize where we are able to assist and to help you out. So Nelson Labs is definitely able to help you with the design of a cycle, the material packaging evaluation, and the heat iteration testing. They can definitely help you with historical data and a file review. We have experts that can give you training on the standards and the revision of the standard, because currently the 11135 as well as the 10993-7 are being revised. and we can definitely assist you to find the best PCD design and selection of the location where we are going to put the biological indicator. Nelson's sister company, Sterigenics, is also having a group of experts, and we can definitely help you to have this best cycle optimization as well as the validation optimization, and work with a PCD that has been optimized at Nelson to make it work in the industrial chamber. So just to summarize this second portion so that you have a bit of time for questions, we have to think about reducing the gas concentration, but maybe recalibrating the BR resistance and adjust the heat duration time to have a faster turn time is really something that we are looking for when we have this 3R program. The benefits that we will have at the end ultimately are, of course, we are still able to continue to use EO because it is still today sterilizing 45% of all the medical devices that are sterilized today in the industrial chamber in the US. And we need all these devices to be available in hospitals. We can decrease the residues so that we are limiting any potential risk for the patients, but also that we can reduce the heat duration time so that we can have a faster turn time and faster product available to the market. If we are able to combine different factors that can improve the cycle duration, we can also have more capacity, because I know the capacity for ethylene oxide sterilization is always an issue. We have less and less chambers available, and ultimately we can reduce fugitive emissions to really enhance health and safety for workers and communities. So I want really to thank you, and yeah, we have a bit of time for the questions now.
Thank you so much. Yeah, we're going to fit in just a few of the many, but I'll get to how we'll handle that after. But before we begin the Q&A, I'll very quickly run through, if you could, there's a webinar survey on the right side of your presentation window. If you closed it, you can reopen it by clicking on the icon at the bottom of your screen. And thanks for filling it out, because it does help us serve you better. Now, let's fit what we can in here. The Q&A portion, you already know how to put questions in, it looks like, because we've got so many. But you type your question into the text box and at the right of the presentation window, or click the Q&A icon. If we can't, this is the critical part. It's worth putting the questions in, because if we can't answer all of them during today's webinar, we'll be sure to share them with our speakers who can reply to you offline. And let's move right on. I'm just going to have to pick a few of the critical ones. Can you speak about the impact of moving to a new manufacturing facility on the established radiation sterilization process validation?
So that says radiation, so I'll take that. So with moving from a different manufacturer, that's actually something that we've been seeing quite a bit. So with global supply chain shortages, with COVID, with employee shortages, moving supply chain locations, or even just switching suppliers from one down the road to another. So with that, you don't necessarily have to entirely revalidate. There are some options. So one of them certainly is to revalidate. And if the new manufacturer has more controlled bioburden, for example, you might be better off revalidating, so you can, what we refer to as, realize a lower dose. But also there are options to do what is known as a family grouping, and family group the manufacturers into the same sterilization validation family and dose audit family. And that's something that we can certainly help with as an expert advisory services offering. We can help you basically figure out what testing you need to do in order to do such an adoption, and document the adoption, do the assessment, things like that. So there are options other than just revalidating that we can certainly look into.
Okay, let me jump right in with another one that I think applies to a lot of people. When doing residuals, EO and ECH, how many samples are needed for the testing?
Yeah, that's a really good question, because currently the ISO 10993-7 is not giving you any guidance on how much samples you would need, and also not where to place these samples, because we have to be cognizant about the fact that if we have samples on top of the loads, they will be outgassing faster than the one that would be in the center of the load. So we are recommending, in terms of reproducibility and repeatability, to have a minimum of 3 samples that tested by time point, so that we have at least, I would say, the bare minimum statistical relevance with 3 samples. But today there is no regulation forcing you to select a sample size, and I would say 3 is good practice, because you can assess whether or not you have an outlier in your testing and if it's robust enough.
Mm-hmm. Well, we'll go just a minute over, and I'll sneak one more in for everyone. And again, we will forward the questions so you can have them answered. So the other questions, similar to further it, someone's looking for insight on how EO sterilization leads to generation, and how ECH generation can reduce that by controlling EO sterilization process.
Yeah,
I read that.
I'll take this one too. Yeah. So ECH, ethylene chlorohydrin, is a byproduct of ethylene oxide sterilization that is being formed when we have a presence of chlorine, free chlorine, or hydrochloric acid. So it means that a PVC tubing can lead to ECH formation, but when it turns to ECH levels, most of the time it's when products have been bleached to decrease the bioburden, where we could see the most important peaks of ECH. So this formation of ethylene chlorohydrin is taking place already during the sterilization in the chamber, but can still continue during the heat iteration process. Ethylene oxide is a non-volatile component that is dissipating naturally, I would say, because it has a boiling point at 51 degree Fahrenheit or 11 Celsius, where ethylene chlorohydrin has a much higher boiling point. So it has a boiling point at 131 Celsius or 267 Fahrenheit. So it means that it will not naturally dissipate. So in order to prevent ECH formation, you need to check in the raw material the potential presence of chlorine, and try to get rid of it. So that's the only way to secure it, because I would say the dissipation of ECH is not following a kinetic logical behavior like EO is. It's more, I would say, not really totally under control, because it will not naturally dissipate. But formation can occur more likely in acidic pH. EO sterilization is leading to more an alkylic pH, so we have pH around 8, but if we are getting closer to 5, we can have an increase of this formation. So it means that we being cognizant about this, so we can try to identify what would be the chlorine sources that can lead to ECH formation with you.
Okay, as much as parting will be sweet sorrow, let me say thank you. That's all the time we have today. Again, I'm guessing you'll get to those to the questions we didn't answer. We'll be able to help everyone out, because there were lots of good ones. That's all the time we have today, but thanks. We appreciate your time and expertise on the topic. And I'm going to read this fast so we close out. Thanks to Nelson Labs, our sponsor, as well as to everyone in the audience, and also, of course, to Sotera Health. And thanks for your participation, attention, and those questions. And within the next few hours, you'll receive a personalized follow-up email with details to a link to the presentation on demand. So anyone who needs this Focus on Fundamentals Day 1 to watch it before tomorrow and maybe jump on tomorrow, you can invite your colleagues and peers who weren't able to attend now or later as well. The webinar finally is copyright 2022 by Informa. The presentation materials are owned by or copyrighted by Informa Markets. The individual speakers are solely responsible for the content and opinions. And on behalf of our guests, Gregory and Bryce, I'm Bob Sperber. We look forward to seeing you again tomorrow. Whew, we did it. Enjoy your day, everyone.